Okay, well, hi, everybody. Thank you so much for joining us today. I'm really excited to talk to you about the newest instrument that we've just launched at Biolog called Odin 8 and our whole portfolio of tools for doing cellular characterization and microbial ID. So I'm sure you've heard the expression, you are what you eat. And I think that is, one of the best kind of macro multicellular representations of that is this flamingo that I have in the picture. So baby flamingos are gray, as you can see, and as they eat the algae and the brine shrimp that they like to eat, their bodies metabolize the orange and red pigments that are found in that food, and it turns their feathers pink. And so, this is, of course, the phenotype that flamingos are most famous for, but it doesn't really have much to do with genetics at all in this case. And indeed, we know that characterizing phenotypes of an organism can offer a much more comprehensive view of the functional traits of that organism. And, I'm sure we've known for a long time that it doesn't work that where one gene results in one phenotype. Things are much more complex than that. And your phenotype is merely influenced by your genotype. So there's all of these environmental factors like nutrition in the flamingo's case, temperature, stress, all of these different environmental factors that affect your phenotype. So while the genotype is inherited from your parents, your phenotype is not. And of course, there's lots of traits that are controlled by many genes, which really makes organisms more robust to mutations. This is especially true for traits that are important for survival and reproduction. And so we really believe that phenotypic information gives you a more comprehensive view of all of these meaningful functional traits of a cell or an organism. And so while the genotype alone can't tell you what really is happening in the real world, if a microbe is growing in a certain environment and there's other bugs competing for its food sources or producing specific metabolites, what you really need to know is how it's responding and what the phenotypes are. And that's really what our technology at Biolog focuses on. Kind of at the core of this technology, we look at what cells can metabolize or eat. And the food sources in this case are chemical substrates that are made of these basic building blocks. And I'm sure you know that breaking down these macromolecules is controlled by enzymes. And many of these enzymes need cofactors like NADPH to turn over their substrates. And so, one of the ways that we can measure phenotypes of cells is to add a redox reporter dye that is permeable into the cells and it's colorless when it's oxidized. When it gets reduced during any of these metabolic processes, it irreversibly changes color. And so, we can measure this color change to be able to answer all of these different questions. So, what are cells eating? We can do this by putting them in a very minimal media and giving them access to only one substrate at a time in each well of a plate. And we can ask how much of it can they eat? We can do this by making quantitative measurements of that dye reduction. And then how fast they do this. So we aren't just taking single reads, we're measuring all of the data kinetically to be able to get this full picture of many substrates all at once. And so here I'm showing an example of one of our sets of plates, one of our panels, and we call these the phenotype microarray panels. And we have these comprehensive sets of plates that have been specifically designed for profiling different types of cells. So you can profile bacteria or fungi or even mammalian cells. And you can probe a lot of different pathways at the same time. So you can see many of the substrates here are, specific carbon sources, nitrogen sources. There are inhibitors like antibiotics or antifungal compounds. And then there's combinations of conditions, like you can look at specific amino acids as a food source at a low pH or a high pH. And so this example set of 20 plates, it looks like a lot of data, but that's where our instrumentation really comes in to help. That's what I'm going to be talking about a bit more. So with our instruments, this becomes a very quick and straightforward experimental setup because these plates are all pre-filled with all of these different compounds. And so you just put the same cells into every well, you put them in the instrument, and you just let it go. And here is our Odin family of instruments, and these allow you to do all of those phenotypic experiments that I've started to talk about. So, with any Odin system, you can incubate, you can read, and you can analyze all of the microplates that Biolog makes. And you can measure cell growth with traditional optical density measurements, or you can actually measure the formation of that redox dye to look specifically at cell metabolism. And with these two orthogonal measurements, you can really quickly home in on specific conditions that either impact metabolism or growth or both. And something also nice about this technology is because you don't have to start with a pure culture, you can actually analyze phenotypes of a community of microbes. So, for example, how they might respond to different perturbations in their environment. And then finally, you can use the same phenotypic characterization method to actually identify unknown microbes by taking the pattern that they produce on our special plates and matching that to a comprehensive database that we've built over the years of thousands of species of bacteria and fungi. And I'll talk about that one specifically a little bit more later as well. And so Odin 8 is the newest member of this family. It was on that family slide, but what we're excited to be launching today is it's smaller than our Odin L instrument. So that one has room for up to 50 plates at a time, and it's really perfect for labs with multiple users and a lot of samples. But this one, we've really designed Odin 8 to be more accessible. So it's smaller, but equally powerful, and it's more affordable, and it's really meant to meet the needs of labs with lower throughput. So this is what we'll be showing you a couple of stories today of how you can use this to do some of this phenotypic characterization. So for the first part, I'll just talk a bit more about characterizing phenotypes of microbes. All right. So, as I mentioned before, phenotypic characterization is really complementary to genetic profiling. So, this really helps you assess the true fitness of a strain or an organism in a way that if you just sequenced it, that wouldn't tell you. So, for each one of those thousands of conditions that we have available in that panel of plates that I showed you, Odin will automatically read and generate these kinetic curves. I'm just showing you one example here, but it will do this for every well of every plate. And then it can compare them to each other. So it makes it really quick to see which conditions show the same behavior for the cells you're comparing, because those curves would overlay right on top of each other. But in this example, you can see a case where they're not overlaid. So culture A, in this case, had a shorter lag time on this one particular substrate. And so it's better suited to grow on this food source. But then both cultures actually reached the same endpoint, the same plateau at the end. And in this case, the y-axis could be optical density to measure cell growth, or it could be dye reduction to measure cell metabolism. And we're looking over time. And so this here is essentially the same story, but instead of that cartoon graph, this is real data that we collected on an Odin system. And here we're just showing you, all of the little time points represented by the dots on the graph. So for these, here we're showing two substrates. So this is just two wells of a plate. And we grew two different strains of E. coli. One was a wild type strain that's used in the lab, and another one is a pathogenic mutant strain in pink called mutant. And so both strains here can metabolize substrate A equally well. You can see they're basically on top of each other. But for substrate B, the mutant strain shows a much shorter lag phase, and the curve is shaped very differently. So if you had only made, one or two measurements over the course of 24 hours, you would miss this difference in the behavior because you would assume probably that, they ended up the same as each other. But these kinds of differences can have really big implications if you're trying to, model growth of an engineered strain, or you're trying to figure out which species could outcompete another one in a community. So it's really important to have this full kinetic set of data. And so to show you how this type of data can be used in a really practical way, I'm going to turn to the budding yeast, Saccharomyces cerevisiae. And so you probably know this has been used for thousands of years by people to make bread and wine and beer. and all kinds of delicious things. And so over time, we've exploited this natural diversity in different isolates of the same species to be able to find, ones that make the most delicious beer, for example. And then more recently, we've been able to use more modern techniques like mutagenesis or directed evolution to be able to introduce even more genetic diversity. And so, if you want to select a strain of Saccharomyces cerevisiae for a very specific purpose, like maybe to express a recombinant protein, somebody might be looking for a specific phenotype. So, you might want that strain to be able to use a specific carbon source or to be able to use a wide range of sources of food. So for this experiment, we selected 4 different strains of yeast, and you can see some notes about them in this table here. So the first and the third strains are lab strains, and these were really developed to be science tools. So one of them was kind of engineered to make recombinant proteins, and then one of them is used a lot for synthetic biology. There's like a whole library of knockouts made on this strain. And then the second one on the list is a strain that actually has probiotic properties in humans. It's known to improve your gut health. And the last one is a commercially available strain that's used for making wine. So we figured that there should be some differences in the ability of these 4 strains to utilize carbon sources because they've really been selected for quite different purposes. So, to show off the throughput of the brand new Odin 8 system, we picked just one plate here. So, this is the first plate of the PM panels, PM1. And this one has a collection of 95 different carbon substrates on it. So, these range from simple sugars like glucose and galactose, all the way to some more complex substrates. So we took each of these 4 strains, we just inoculated each of them onto a PM plate, and we did it with and without our redox dye. So that's a total of 8 plates, which is just one run of the instrument. And we just left them in the instrument for 24 hours, and Odin 8 did all of the incubation and the reading at 30 degrees, and then gave us the data at the end. So if I blow up some of the small plate wells from the whole plate that Odin software gave us, you can see the 4 colors that represent each of the 4 strains to the left of the graph here. So we actually found a total of 7 substrates that were able to show good log phase growth in at least one of the strains. So in this set of graphs, I'm just showing 3 of them here. So you can see glucose, mannose, and fructose, and all 4 strains are able to use these sugars as their sole carbon source. So remember, they have nothing else to use except this one that's listed. And then I'm also showing that none of the 4 strains could grow on arabinose or lysose, which isn't too surprising. Yeast are generally, more prone to metabolize hexoses and disaccharides. So these, you can see kind of the same phenotype of all 4. So now here's a few wells where we could see differential utilization of some of these sugars by the different strains. So the 2 lab strains, the orange and the black curves, are not able to use maltose at all. And you can see that the probiotic strain and the wine strain could use it really well. And then you can also see that the probiotic strain could not use galactose at all, but it was the only one that could use sucrose really efficiently. The wine strain could use sucrose somewhat, and it was the only one that could survive on maltotriose alone. So you can start to see these, really interesting differences between different strains of the same species. And what's also interesting is there's previous research that was published that shows that this probiotic strain, the one in green, um, is not able to use galactose at all. So that had been previously published, and it was actually initially characterized as a completely separate species, partially because of this phenotype. But then later it was, it's now accepted that it is actually just a subspecies. So we were able to reproduce this finding that, it cannot use galactose at all. And we did so in this plate with dye and without dye. So because we're only looking at carbon sources in this plate, the growth was really tightly correlated with metabolism. So we didn't see any differences between those two. But we could expand this work to a larger set of plates And then we could see a different story with some other substrates. And I'll actually show you that next. So in this set of data, now we're looking at just one well of a PM plate. But on the left, we're actually breaking it down to look at just optical density with no dye. And then on the right is this metabolism assay with the redox dye. And you can see here that, again, we're going back to our E. coli strains here. So this is the wild type and the mutant strain. And you can see that the metabolism graph looks, very, very similar. So they're both metabolizing the substrate at the same rate. But the wild type strain is able to grow or divide more than the mutant strain. So this is really an interesting finding. And there's lots of examples out there that show that growth can be impacted, but you can kind of persist and maintain metabolism. So some microbes have preferred food sources for doing things like making their cell wall that they need to divide. And the inverse can be true too. So we know that there's cases where organisms can undergo a metabolic shift without changing their growth rate. So there's studies done in fungi that show that different cell phases can actually have different metabolic profiles, even though they show the same doubling time. So this is a nice way that you can really separate these two things and look at, both at in the same experiment. And when you're trying to make a kind of comparison like this, Odin software actually makes it even easier for you. So you can just tell the software which sample is your reference sample, which in this case would be our wild type, lab strain of E. coli, and then which sample is your test or experimental sample, which would be the mutant strain. And then what you can see is that everything in this kind of blue-gray color is the overlay of the two samples. But wherever you see a significant pink or a significant cyan burst of color, then your eye can immediately see that there's this difference between the two samples. So when you're looking at many wells of many plates at once, you can, see that really easily. And then, of course, Odin goes further and calculates all the parameters like the area under these curves or the maximum height or the average height. So you can actually quantify these differences as well. But when you're just scanning through your data and you want to see what's going on, it's really easy. So here's a snapshot of some real data from just one plate again, and this is run with dye and without dye. And this is actually our 11th plate of this series. So this is PM11. And this one has different drugs or inhibitor compounds. And each drug on this plate is put in a series of 4 increasing concentrations. So on the bottom, maybe you can see these triangles that represent the series of increasing amounts of drug. So if you quickly, glance at the data, you can see some compounds where both strains are essentially the same on all concentrations of a given drug. But you can also see some differences where, maybe the difference becomes apparent at a particular concentration. And then one of the compounds on this plate that I've put a box around is enoxacin, which is a DNA topoisomerase inhibitor. This used to be used as an antibiotic, and it prevents the bacteria from replicating their DNA. So on the bottom, you can see this is the plate grown with dye, and you can see that at the lower concentrations of inhibitor, both strains are basically mostly overlaid, right? So there's no real difference here. And then at the 2 highest concentrations, neither strain can survive. But if we look without dye at the top, you can see that the pathogenic strain is not actively dividing or growing. It's just using its energy to stay alive or persist, whereas the other strain can actually keep growing in the presence of this drug. So, and then same thing at the high concentrations, neither strain can grow or survive. So this is an example where, you can use this data to look and make comparisons very quickly. And so getting all of this data for the same samples is really useful for lots of different applications. But one of them is, if you're trying to optimize a fermentation process. So here's, again, a set of 20 plates, and you can see that each well is really probing different questions. So you can interrogate your microbes to see maybe which ones are adapted to high salt conditions or heavy metals, different pHs. And then this can help you make a call about how fit or robust a strain is under a given set of conditions. And I should also mention that this set of plates is one of our standard set that we make, but we can also work with you to customize plates if you have your own particular conditions or applications that you want to look at. And so for these types of applications with fermentation and bioprocessing, it really comes back to what the bugs are eating, right? So if you're feeding the organisms something suboptimal, then they will be in a stressed environment and they will not behave the same as they behave in an environment where they're not deprived of their ideal nutrients. And then, like I mentioned, you don't have to run a single pure culture on our plates. There's a ton of really interesting publications that have used our plates to study communities. So, you can imagine lots of use cases for that. So, if you have bacterial respiration competing with fungi, for example, you can do a lot of really interesting work there. And if you're trying to make a product with fermentation, you probably care a lot about trying to make sure that it's either respiring more than growing. Like if you're making beer, you don't want the yeast biomass to increase too much and foul your beer. But if you're making a high protein food, like a meat substitute, you might want a biomass rich fermentation. So you can use this in so many different ways to monitor your production strains. Something else I just wanted to point out is you can actually generate a kind of a fingerprint for your strain to make sure that it's not being contaminated with something else, and also that it's not drifting significantly as it repeatedly gets passaged over generations. So a lot of really important use cases for monitoring all these phenotypes at the same time. And then even though, we're talking about our new instrument, Odin 8 today, I just wanted to put in a plug for the larger system, which is Odin L. And this one really makes it easy to scale up. So rather than just looking at a few plates at once, you can look at so many conditions at the same time. So here's just one example of an experiment where we ran a whole set of 20 plates for two different strains. So that was a total of 40 plates, or at the top it says 3,800 conditions. And one of the strains here is the wild type E. coli strain. And then the other strain here was something that had a deletion of the MALF gene, which is part of that maltose transport system that the bug needs to be able to use maltose as a carbon source. So on the right, you can see that those are the two curves that show that, yes, the mutant strain has lost the ability to use maltose and maltotriose, which comes in by the same transport system. And so all the conditions we're comparing, these two that we've colored in pink really stand out as outliers, right? And then at the same time, we saw another phenotype stand out that we've colored in blue. And here there was a gain of function for resistance to some tetracycline family of antibiotics. So the mutant here is able to be resistant, whereas the wild type strain was not. And this actually makes sense to us too, because we know that there was a chlorotetracycline resistance marker that was actually inserted at that MALF locus. But of course, if you did not know what to expect in terms of what genetic differences had been made, if something had been randomly inserted, this is a really fast way to be able to screen through all these conditions and quickly find significant differences between any pair of strains. And of course, so many pathways are interconnected. So even if you think you know what effect a mutation will have, it's really nice to be able to screen all these conditions at the same time so you can identify these other outliers, right? So there's a few other things here that we could look into and see what those are. So this is a way to do a lot more at once. And so just before we move on to the last story, I wanted to show a couple of screenshots from Odin software and just highlight that it really makes it easy to generate reports from all of your data. It makes it easy to export all of the raw data and the processed data in different formats. And of course, it knows what compound is in every well of the plate. So it generates all these tables and graphs for you. And we are actually continuously adding new features to the software. So as an example, I'll put a little plug in for an upcoming webinar on the community analysis features that we've been working on. So stay tuned for that one. And with that, I wanted to just talk a little bit about how you can also use Odin 8 to identify unknown microbes using the same concepts. So, who needs to do this? There's a lot of different applications where it's important to be able to quickly identify contaminants in your manufacturing space or, anything growing where it shouldn't be growing. So, this can be all kinds of different products that you're trying to make or in monitoring the environment, like water or wastewater. So, lots of different people have been using this technique. And we have these specialized plates that are, specifically designed for doing ID. So each of these four that are shown below are single one plate universal test panels that we use for identifying specific categories of microbes. So you can see we have a plate for bacteria, and that encompasses gram negative and gram positive bacteria. You actually don't need to know which one it is. We have a plate for anaerobic organisms, a plate for yeast, and then a plate for molds and filamentous fungi. And so each plate has a unique substrate or chemical in every well. And so what you do with these then is the user just takes an unknown microbe that's been cultured or streaked out on a plate. You just dilute it in our minimal media and you aliquot it into every well. That takes about 1 minute. And then you just incubate it in Odin and it goes. And in every well, if the organism is able to use that substrate to grow or metabolize, then the dye changes color. And then in some wells, there's inhibitors. So, same thing there. If the organism can survive in the presence of the inhibitor, you will also see the well change color. So, these plates have been designed in a way that each organism at the species level will show a unique pattern on this combination of compounds. So, it's kind of like a distinct fingerprint. And at each interval, Odin reads the plate and tries to match the pattern with something in our database. And so, what's in our database? Well, we have a lot of different species here. So, you can see about 1,500 species of aerobic bacteria. We have, I think, 361 species of anaerobes, a lot of yeast, and then over 710 different filamentous fungi. So, this is a really powerful database because of its relevance. Filamentous fungi in particular can be really tricky to identify with other methods. And we have this large number that are very common in the environment, in air and food and soil. So, to be able to use this for environmental monitoring, all you have to do is just take a swab or a swipe from different areas, or even from your raw materials or your final products. And you can use this to identify and mitigate contaminants quickly. And so for some of this proof of concept work, we took our four different types of ID plates and we ran them with a selection of different microbes here. And using Odin, we were able to identify this range of species just as we would expect. This has been around for a while at Biolog, and so we're just kind of showing that Odin can do this ID just as well. And this is really a complementary way to other methods of identifying microbes. And we have this database of almost 3,000 species in total. And we're continuously expanding the list with new species and strains within it. And I think I had this on a previous slide, but if you're working with novel proprietary strains, we can also work with you to build a custom database as well. So with that, hopefully you've seen some data that shows how Odin and our newest Odin 8 can make your life easier and give you a lot of rich phenotypic characterization data on any cell or organism that you're working with. So as opposed to something like a plate reader where you perturb your cell's growth by taking them out of the incubator every time you read them, and you'll miss all of that important data that happens in between those reads. These are dedicated instruments that go with our user-friendly software and our plates that all really work together to give you more robust and actionable data. And so now that we've introduced Odin 8 to this family, you can see that the only real difference between the instruments is the plate capacity or the throughput. So all of the other features and software are the same. You can configure both systems to do microbial ID, you can do phenotype characterization, and now community analysis as well. And all of the other sort of specs are the same. And I'll also just mention that at Biolog, we have a whole ecosystem to help researchers characterize cells, whether that's microbial or mammalian, like I didn't talk about today, and to identify microbes. And we do this with this whole range of tools here. So our fit for purpose plates and the chemistry that I talked about. We have the suite of instruments for any throughput that you need, and they are all powered by our software and our comprehensive database. And we also have a lab service and testing facility that's ISO certified. So, as well as doing this phenotypic characterization work that I talked about today, and the ID, this group can also do genomic sequencing, MALDI-TOF, enumeration, and lots of different lab services that are very relevant to anyone working with microorganisms. And finally, Biolog and our core technology, we've been around for a long time. It's actually our 40th birthday this year. We have instruments installed and in use all around the world, 6 different continents, and we have over 5,000 publications that cite the use of these products for many super, interesting and different applications. And so we're really excited to see what comes next and what Odin 8 will empower in terms of new research. So thank you so much for your time today, and I'm happy to take any questions that we have time for.